Now, I have the privilege to invite Dr. Rohit Bishit, who is a scientist of regulatory toxicology working with PETA India. He did his PhD from University of Auckland, New Zealand, and his postdoc from BITS Pilani. He has completed numerous projects from AIMS New Delhi and University of Kansas, USA. His research work has been recognized by American Association of Pharmaceutical Scientists. And now I invite Dr. Rohit Bishit to enlighten us on humane relevant methods in drug development and safety testing, where we are and what's next. Dr. Rohit, over to you.
Sure. Good evening, everyone. So, in today's talk, I will share the information about non-animal methods and what significant changes is happening on global regulatory bodies, what PETA India science team is doing globally and in India for the advancement of non-animal methods, what Indian regulatory bodies are changing in regulatory guidelines regarding the non-animal method acceptance, and what type of programs or initiatives we are working or running in India for promoting non-animal method. And of course, at the end, the most important, the challenges and opportunities which relates everyone, industry also, academia also. Because with non-animal method, it looks good when we talk, but when we do it in practically establishing any technology in our lab, there are some regulatory changes, there are some technical challenges like hands-on training, expertise on handling these, development of these, because if you search any non-animal method, whether it is organoids or organ on chips or tissue culture 3D models, regulatory bodies approve them. They are integrated in OECD test guidelines, but most of the models are commercially available in abroad, in Western countries. In India, now we have started developing these organoids, organ on chips, but they are at initial stage. We have to validate them. And for validation also, there are a lot of requirements from regulatory bodies. So these are in India. So in India, this first one is hands-on experiment challenges. Second is the funding, available of funding for the development of these alternative models in India. So let's begin with some introduction of non-animal method, then slide by slides, we will talk about different aspects related to non-animal methods. So initially, I will just highlight about some limitations with animal model. Then these limitations are endorsed not only by the scientists, but by renowned scientific agencies like NIH. And as you know, like 90% of drug which shows the promising results in preclinical trials fails in the clinical trials. And it has been investigated the results concluded that about 18 to 89% of research are reproducible. So reproducibility is a lot of challenges there when we talk about academic research also. So this is also a concern with the animal testing. And according to some literature, has been found that only 10% of highly promising studies enters into the clinical trials. And even though the failure rate is more than 95% when we consider about some certain disease conditions like Alzheimer, still there is no alternative model. We have to use the animal models. Like cancer, HIV vaccine, strokes, and sepsis, these in vitro models are still in the R&D phases. So our ask with the regulators and with the scientists that whenever you are working with the animal model, if there is an alternative model which can be used, you should shift from the animal models to the in vitro models, or do a comparative study, and if it justify and you feel confidence on the results, you can transform from the animal model to non-animal model. And in the areas where there is no availability of in vitro models, then of course you have to use the animal model. There is no other options. Considering this, we have the PETA scientists have developed this document known as Research Modernization Now. This is a roadmap for how industry or academia and regulatory bodies can shift from the non-animal models to in vitro models. So in this, we have discussed about the different type of animal models, the limitations, different sections of biomedical research, cardiology, cancers, cell gene therapies, new upcoming therapeuticals categories, what type of animal models are available, what limitations are there, and what are the in vitro models which are available there which you can use. And what the regulatory bodies are talking about these in vitro models. And then we have developed some strategies how you can shift from this animal models to in vitro, because this transition is not easy. It takes time. It will not happen in one day for the regulators also and for the industry also. To change its whole quality management system in a small time is not possible. So we are starting from the ground, from the first step, spreading the awareness, hands-on trainings, and then slowly communicating with regulator and so on. But the main objective with this is that replacement of animal method with the non-animal method in the areas where animal models have already shown to a poor surrogates for humans. Critically reviewing the additional area of research to determine whether animal use can be phased out. So if you go to the website of Animal Controlling Authority in some countries, they have database. You know, like how many animals has been used in 1 year for different projects. We are communicating with CCSE also, DBT BIRAGs also, that we should have developed such type of database, like what type of categories of research there, where the proposal is received, what type of animal models this year we have requested for, animal requested for, and ultimately after that we have to also enter the details about what is the output, because the next person who want to use that models will see what either this animal model is having some significance for the studies or not. So it will save a lot of animal lives. Then redirecting funds. So we are in communication because we work with the regulators, we work with the scientific agency, we work with the industry, we work with the academic and industrial scientists for the advancement. So we are continuously asking with the DBT and other scientific agencies to, increase the funds or allocate a separate funds for the non-animal development. So recently DBT has announced a separate funding opportunity for development of in vitro disease models. And now we are also planning to develop a center of centralized facilities for the development of in vitro models. So introducing ethical harm benefit analysis, this is also in process. And education training, this is the most important part, which is we are doing with the industry also, with regulators also, and with the academic scientists. So last time we have sent about 7-8 Indian scientists to Institute of In Vitro Sciences, US, for hands-on training on different in vitros. And next 2026, we develop a collaboration with the National Institute of Biologicals, where we are organizing this hands-on training for industry and academic scientists in collaboration with Institute of In Vitro Sciences, US. So starting with the drug development, this is the project which I am leading in India, equine-based therapeutics. With the equine therapeutics, there are some risk of mild allergic reaction, serum sickness. There is a risk of transmitting of microbes, which is very important or critical when we are in pandemic conditions. And there is a batch-to-batch variability and short plasma half-life. That's why when we discuss about the equine-based therapeutics, we can't store them for a longer time. So if you see the latest government initiative about promoting the rabies vaccine or snake bites, the vaccine scarcity is there to, in urban and rural areas. So what WHO, because this project is requested from WHO and UK Health Agency, is that they want to shift from the equine-based therapeutics to the recombinant technologies for the old molecules. So we are in India, we are promoting recombinant technologies in collaboration with DBT. They have national biopharma missions where they organize different workshops with the stakeholders to promote recombinant technologies. We are also working with CCSA because they have the equine guidelines, which is 2001, which is not updated. Now they have constituted a new committee and reviewing these documents. We have also had an opportunity to talk with the DCGI, DBT BIRAGs, so that now you will see soon there will be a revised guidelines on this. We have a collaborations with AppCam for the development of monoclonal antibodies. We have a collaborations with AppCalis. We also working with US FDAs in developing the old molecules into new recombinant molecules. We do RN research and developments. We have developed recombinant diphtheria antitoxins because the available old one is equine-based. So this is the project which is supported by UK Health Agency and WHO. So in animal testings, we have different type of non-animal methods, reconstructed human tissue models, spheroids, organoids, bioprinting, precision cut organ slice. We have cell-based assays. We have in silico models. We have now organ-on-chip models, where we have a single organ, multiple organ models. Now we have combination models also, in vitro coupled in silico, organoids, inner organ chip models. So These are the models which are commercially available. These are the leading company, MedTech, IPSCON, which have regulatory approved model in vitro method, which company and regulatory bodies are using. And also Indian companies are also right now using this, and they are integrated in OECD test guidelines. So I'm just giving you the slide highlights about, and then I will go with the main topic about regulatory significant changes. So this is spheroids. This is organoid, but the important thing with organoid is that in 2022, US FDA has granted first approval of the use of organoid and organ-on-chip technology for new drug approvals. 2025, US NIH announced a reward of 87 million in contracts over 3 years to standardize organizing modeling center. And this is the curator, they having their organized platform. So they have got the first US FDA IND approval for the anti-cancer drug, which is tested and on organoid models. So this is how the regulatory body is also accepting the alternative models. So this is just an example of an organic model by Cherry Biotech, how they have this, which can easily use in a lab, a small setup, and different areas where they can use. Biochem printing already installed in different academic institutes in India. We have in MIT, NIPERS, IITs, they are using. In this, you can use it with different type of cells. You can choose the organ. Then, depending upon the cell, you can develop these 3D tissue models. And depending on that, your project, you can, take a lot of information from that. So in different areas, they can be used. In silico models, we are the one, we have developed first regulatory approved QSR toolbox, which is known as OECD QSR toolbox. We have a collaboration with OECD on different projects. This is one of the projects, and this toolbox is already OECD approved and freely available for the use. US FDA also accepted this PBK simulators for the pharmacokinetic studies of the drug molecules. And this in silico models, we have different, but recently they have also used for reproposing of drugs which are very much used. Even in the COVID-19 cases, we have remdesivir, which is reproposed for its use. Organ-on-chip models, now we have organ-chip models for each and every organs. And I would like to discuss about this liver-on-chip model, because this is the first model which is accepted by US FDA for its iStent pilot program. So US FDA regulatory bodies have their separate iStent program where they, depending upon the technology potentials, they accept this proposal and collaborate with the stakeholder for the advancement and whole study and approval of this model in a large scale. This is another models which you can use, precision tissue slice models from the diseased person, and then you can use this for the drug screening. Now, this is the important section I want to discuss about global regulatory shift. So first, I will discuss about the US FDA. So US FDA have from time to time, so I'm highlighting the recent changes. There are a lot of changes. If you go to the US FDA website and type non-animal testings or alternative testing, they have different changes, modification, guidelines for each and section. Monoclonal antibodies, vaccine, veterinary vaccines, pyrogen testings. So time to time they release different type of guidelines. Recently they have FDA Modernization Act where they have accepted these all in vitro methods for the preclinical testing of new investigation drug. Following this, India has also updated their new drug and clinical trial rules. Now this amendment has already approved by the government. They have included all in vitro models, in silico, in vitro, everything in the guidelines for the investigations, preclinical investigation of new drugs. So this is another recently happens. The FDA announced the plan to phase out the animal testing requirement for monoclonal antibodies. This is still open for the stakeholders. So we are also proposed some of the tests for the recombinant monoclonal antibodies are in vitro and in silico coupled method, which we have proposed for monoclonal antibodies for immunogenicity, specificity, and tissue bindings. Because for the immunogenicity, the rabbit model, which is used by the previous old guidelines, ICH, it is already known that it is not good model for immunogenicity investigation. FDA Advanced Drug Development Innovations, iStent, is a program for which For medical devices also, if you go to the MDT program, this is a separate program started by the FDA where they have accepted in vitro models for the investigations of the effects by using in vitro model. We are working with the FDA on the MDT. We have used monocyte activation test. We have validated this test for medical implant, tooth implant basically, and we submitted our validations data with the US FDA and it is in discussion. So these are the different guidelines for an alternative for their validation, qualification requirements. So a lot of changes is happening in US FDA. US FDA also do the collaboration work with the stakeholders. They have developed the comprehensive in vitro polyarrhythmia assay, which is, it's a big study concluded, and now these are included in ICH guidelines. They have also did the cross-cutting FDA applied research, liver microphysiological systems. They have also working with the stakeholder for 3D model for lung airways. So inhalation toxicology, a lot of things is going on in inhalation toxicology in US FDA and European pharmacopoeia. There are different type of in vitro models which are already approved for inhalation toxicity. So this is about European pharmacopoeia. A lot of things are going on in European pharma. Even though for non-animal toxicology or non-animal testing, European Commission is ahead of all the regulators. So they have completely removed abnormal toxicity test. They have also removed the in vivo methods by in vitro methods for quality control of vaccines. They have replaced the histamine sensitization test for vaccines, toxin. Now they are decided to remove all the rabbit pyrogen tests from the monographs. So I will go to the India. So what we do, we work with the regulator, policymakers, scientists and industry for advancement of non-human methods. These are the different projects that we are using at different countries. We work with the regulators, policymakers on different areas, providing awareness, hands-on training, communications with the regulator, proposals, validations, supporting industry for OECD test integration programs. So these are the recent changes which has happened in India. These are small changes. Complete removal of, if you go to the Indian pharmacopoeia, the regulatory books, they have completely removed the abnormal toxicity test. Target annual batch safety test is completely removed by the 13 veterinary vaccines. New drug clinical trial amendment, I'll already discuss. New revised guideline for biosimilars. So we work, biosimilar manufacturers of India contacted us on provide us the authority where we have communicated with DBT committee and CDSCO. Now this revised guideline where they have waived off the requirement of animal test for the market authorizations of biosimilars. And these are the different others. So other than this, these are our initiatives. We do non-animal method focused sessions. We provide hands-on trainings to the scientists. We also award in equipment or instruments and grants, research grants, and all this. Yeah. Thank you.
Thank you.
Yes, this is fine. Yeah. Yeah.
So first of all, thank you for giving us a very comprehensive overview of non-animal methods of testing, particularly toxicity testing. Leaving aside the ethical issue, what bothers me is the premise for shifting from animals to other models is that these animal models are useless anyway when you move to humans. Most of the candidate drugs which are showing success in animal models are not succeeding in humans. So where is the guarantee that if you use these alternative methods, they'll be successful in humans?
So for this,
one other thing is that many of these models are going to be proprietary, so they're going to be much more expensive than animals.
So answering your first question is that I'm giving take example of pyrogen testing. So please, sir, you can have a seat. So in pyrogen testing, we are working with regulators and industry about monocyte activation test, which is in vitro test for pyrogen. Initially, in regulatory guidelines for pyrogen, we use rabbit pyrogen test. But where rabbit pyrogen test is not good test because it's having a variability, European pharmacopoeia already removed. Indian pharmacopoeia already removed rabbit pyrogen test from majority of the drug monographs. Now they have included bacteria endotoxin test, that is LAL assays. But the problem with the LAL assay is that first it is depend upon the blood of horseshoe crab. Second is that it only detect endotoxin pyrogens. It can't detect non-endotoxin pyrogens. Non-endotoxin pyrogens is very important when you consider the biological formulations. So now, currently we have only one test, monocyte activation test, or rather rapid pyrogen test for the detection of for the pyrogen, any type of pyrogen. Now, the technology and if challenges in the term formations. What we are doing, we recently had a meetings with all the TMAT test provider, global TMAT test providers. MAT Research is that, MAT Biotech is that, CTL MAT is there, Immunospot is there, Indian providers also there. We take their views. Then we had a separate meetings with the regulators. CDSCO is there, IPC is there, DBT BIRAC is there. We invited CDTL, CRI, NIB is there. We take their views point, how best the way to take these projects so that the transition will be smooth for industry also and smooth for the regulator also. Then we had a meeting with the industries. All the leading bio manufacturers are contacted. Now what we are doing, we have developed a validation protocols. We are doing the validation of monocyte activation test kit. And we do this validation in collaboration with National Institute of Biologicals. We are in collaboration with National Indian Pharmacopoeia Commission
That
also to show them how effective is as compared to the rapid pyrogen test to build a confidence that,
No, that is specific for the pyrogen
Yeah.
test. When you go on to move on higher models like organoids and 3D tissue models.
So for this,
Only
if-
time will tell how successful
No,
are.
because if you consider monocyte activation, just as an example, it is already validated, a lot of literature is there. But what regulator wants is that you have to do a validation, product-specific validation, again in India, in Indian specific condition. Then you have to review data. If I take example of organoid, it's already validated, everything's done. Again, we have to do a specific validation in collaboration with the industry. We have to do the comparative studies with the ongoing models and to show and convince the regulator that this models is giving the same results as compared to the old traditional methods. And then they are agreed to, because unless it comes into regulatory guidelines, nobody will, bother to use them. So most of them are in research base. Yeah.
So our final speaker is Dr. Preeti Sood. She is Professor of Pharmacology at SGR DIMS Amritsar. She actively contributes to research, conferences and pharmacology education, and she is published in national and international journals, including her work on drug addiction awareness among medical students. I now welcome Dr. Preeti Sood to please share her insights on good clinical practices. I welcome you, ma'am.